with cellular therapy. And then also, which is very important for upcoming generations like yourselves in the medical space, is advocacy and development for these therapies. We have a lot more control as physicians in leading the industry and pharma initiatives by promoting for our patients and advocating for our patients on what is needed for future therapies and unmet need for certain patient populations. And we all know there's a lot of unmet need in our patient population of interest. So let's talk about the relevance to specifically cell and gene therapy to musculoskeletal and neurological disease. I don't have to tell this group we recognize the profound impact of disability on society and the growing need for sustainable and effective treatments for our patients, especially as policy changes, as we've seen this past year. It continues to limit coverage for our patients and access to care. Currently, more than 1 in 4 individuals have a disability in the US, making them the largest minority group in the country. Alarmingly, only 1 out of 4 adults with a disability have access to a healthcare provider. The financial burden is also very significant, as we're aware, with quotes about $232 billion spent annually on disability-related costs, and this is only for working age individuals. I wanted to point out on the right side of the slide are all the conditions we treat, or we commonly see in PM&R, and all of these have an FDA-approved cell or gene therapy on the market or a phase 3 clinical trial underway for a potential treatment. That being said, I do want to preference not every patient will benefit from these therapies, and each of these therapies carry significant risk. So when we discuss disability specifically, I'm very mindful of the language we use. Terms like cure or treat can be very sensitive, as disability is often an integral part of a person's identity. However, I believe regardless of the disability, most individuals would prefer to live without painful symptoms or a high risk of mortality. The saying, when you've met one person with a disability, you've met one person with a disability, is especially relevant to the context of novel therapeutics and cell and gene therapies when you could potentially reverse entire pathophysiology of a disease. So I have several questions for you guys to start off with. Besides the unmet need in both the access to care and therapeutics for those with complex disabilities, it's also important to acknowledge the natural progression of the muscle changes with aging. So after age 30, what is the percentage of muscle that a person loses per decade? You guys can throw out any numbers that you think.
Holly in the chat said 5 to 10%. Has gone as high as 15, landing somewhere around 5, somewhere between 5 and 10% is what people are saying.
Yeah, definitely. That's very close. So 3 to 8%. So around 5, yeah, definitely. And then how about in severe disease states between the ages of 70 to 80? So if we have a patient with stage 4 cancer in their 30s or 40s, or a patient who is naturally aging in their 70s to 80s, how much muscle can a person lose within a decade? Any guess?
We have ranging from 50% to 15%.
Close, 20 to 40. So we're all in the vicinity, so pat yourselves on the back. That's really good. Now, what do all these patients have in common? So we have a 40-year-old with terminal cancer, a 90-year-old with critical illness myopathy, a 70-year-old with Duchenne's muscular dystrophy, and a 50-year-old with ALS. This is my last question, by the way. So they all have skeletal muscle dysfunction. They often share, all these individuals or patients, share common characteristics like respiratory issues, compromised immune system. However, at the cellular level, they exhibit skeletal muscle dysfunction regardless of the underlying disease process and its cause. Many of them ultimately have high mortality rates due to complications from disease-related or cardiopulmonary issues. So with that, that is the why of why we care about these novel therapeutics, specifically in the orthobiologic and cellular therapy world. Now, as far as terms, I know the majority are probably medical students, and I know when I was a medical student, I didn't really know how to differentiate between certain terms that we use quite often, and sometimes inappropriately within the musculoskeletal world. So just so everyone's has a baseline going into this presentation, I just wanted to go through different terminologies that we frequently use, and sometimes we use them inappropriately. So frailty is a common word we use, and this is a syndrome of physiological decline, usually and later in life. This doesn't have to mean that there's a co-current underlying condition or comorbidity. Sarcopenia is a syndrome characterized by loss of muscle mass, strength, and performance. It is sometimes referred to as age-related loss of muscle mass. Cachexia is the complex metabolic syndrome associated with underlying illness, usually cancer, but it's characterized by loss of muscle with or without loss of fat mass. And then malnutrition is used to describe a deficiency, excess, or imbalance of energy intake and or nutrition. This results in a measurable adverse effect on body composition, function, and clinical outcome. Now, all of these could exist in one patient. Some may overlap with one another. The biggest issue with identifying patients and truly diagnosing them with the appropriate muscle dysfunction term is that we do not have objective laboratory tests. We do not have a laboratory test where we can say, oh, we know exactly what's happening on a systemic level with the muscle. We can do muscle biopsies, but that only shows us a very, very small picture of what's happening. And I've become friends with my pathologist fellow colleague in the pathology department, as that's where my fellowship department is right now. And the ones that analyze the muscle biopsies constantly say they don't like to do that because it's extremely subjective. There is not a very good way to objectify muscle biopsies and to see what's happening as a whole for that patient. So right now, there is a lot of need for biomarkers and objective laboratory tests within the musculoskeletal world. Now getting into the cell therapy orthobiologics. So hopefully we're all kind of on the same page of the intro on why this is important and the background of musculoskeletal and where there's some gaps there. So overview of cell therapy versus orthobiologics. Let's just start basis at the definition level. So cell therapy has several different terms that you may hear: cellular therapy, cell transplantation, or cytotherapy, which is a term I was not aware of until I was in fellowship. And this just means it's a therapy in which we use viable cells. They're injected, grafted, implanted into a patient to have some sort of medicinal effect. Now, orthobiologics consist of biological substances used to treat muscular skeletal conditions. Now, in the cellular therapy world, I know Will and I were discussing this before, orthobiologics is not considered cell therapy. And that's because the mechanism of action for orthobiologics are, one, not targeted. Usually they're not really fully understood. And there's no standardization for truly understanding what we're putting back into the patient when we are using a biologic substance. So there's not a standardization for cell viability, cell count, and validation for that, along with your general infectious disease testing, genetic testing that we do on cellular therapies. However, I really do see in the future, based on how orthobiologics are treated from a scientific perspective, it could be considered a cellular therapy. But simplistically, right now, you can think of cell therapy more as it's a manufactured standard product. We know exactly what we're getting. We know exactly the target for the patient that is receiving the cell therapy. Versus orthobiologics, we have somewhat of an idea, but there's no validation. Each product is very different from one another. And we truly don't know if we're hitting our target and we're wanting it to do what it's supposed to do each time. Examples between orthobiologics and cell therapies. So orthobiologics can include, but not limited to this chart, PRP, probably the most common that we're aware of, BMAC, bone marrow aspirate, and then NFAT. Cellular therapies for musculoskeletal reasons and neurological reasons that are commonly used right now in clinical trials are mesenchymal stromal cells, umbilical cord blood, and then induced pluripotent stem cells. There are two mechanisms in which cell therapies work. And this kind of can be, I guess, we misinform trainees up and coming, especially when we use the word like regenerative medicine. What does that actually mean? And what is the actual like standardization of that definition and the quote unquote, hopefully soon to be subspecialty of regenerative medicine? So cell therapies in general have two mechanisms that we care about. One is stem cell, progenitor cells, or using mature cells to engraft, differentiate, and have long-term replacement of damaged tissue. So this is the regenerative medicine sphere. And this is like the bone marrow transplant and CAR T therapy that we're using in the hematological world, where we are really trying to cure that patient. And we're trying to eliminate all their disease cells by infusing healthy cells. So that is regenerative medicine sphere. The second mechanism is what we're more likely doing in the biological world right now. Hopefully that will change as research gets better and you guys are coming up and being leaders in the field. But using cells to have some sort of endocrine effect. So we are not necessarily regenerating or having a curative type effect, but we want to use those cells because we know that they stimulate growth factors, chemokines, cytokines. This is where orthobiologics live right now. So you're ahead of a lot of attendings and leaders in the field right now anyways, just by knowing these simple mechanisms of action and differentiating between the certain terms. I will say it is very important that we are able to describe the science to the world appropriately. And we don't have too much hype or too much hope in a sense, because it can cloud the picture and it can cause a ripple effect on whether the science does succeed for patients and access, as well as investors, on whether investors, big pharma, biotech companies want to invest in this science. If it has a negative connotation to it or an inaccurate description that the leaders are using and physicians are using, it can prevent advancements in the field and potentially treatments coming up. So getting into the nitty-gritty details of orthobiologics versus cell therapy. I'm going to jump into cellular therapy. This is the world I am right now, and this is where I feel the happiest, I guess you'd say. It's very standardized. The regulatory oversight is, I would like to argue, more intense than medications out on the market right now in big pharma. It is very much, there's a lot of regulations, a lot of accreditation bodies, as it should be. And so this is the world that I'm feeling comfortable now. So these are the variety of cell and gene therapies on the market or within phase 2, phase 3 clinical trials at this time for a variety of different indications. And this is specifically with cell therapies, not orthobiologics right now. I first want to go over key concepts from a basic level of what really differentiates between cell therapies and orthobiologics and why they are so clinically different right now at this time, even though I really don't think they're that different. So I want to paint a good picture of this. Right now in the cell therapy world, even though we can use all these different techniques, we can use gene editing, CRISPR, we can have induced pluripotent stem cells and differentiate them into neurological type cells. We can have CAR T where we genetically engineer a T cell to attack and target an immune cell or tumor cell. And we can have EVs, extracellular vesicles, where we have targeted drug delivery. We put the drug inside the EV so it's packaged really nicely and it targets and delivers right where we want it inside the patient. All those are very different for sure, but the manufacturing is very, very similar. So the platforms can be different, but the manufacturing developing these therapies are very similar. So we have a source tissue, whether we get fat from the patient or we have them go through apheresis or leukapheresis, meaning we're taking cells from their blood. We transfer that to a manufacturing site. At that manufacturing site, which could be either big pharma or an academic center, we isolate the cell of interest. In between isolation and expansion is the time where we can genetically engineer. So an example in CAR T world is we want to isolate those T cells. So we isolate the T cells and then we want to genetically engineer a CAR. So it's full of an antibody domain where it attacks the tumor of interest. And then once we have our ideas, I'll do our perfect cell and in the genetic engineered cell, we want to expand it, make it into billions of cells. And then we want to harvest those cells to get them ready for administration. We transfer them back to the bedside and deliver them to the patient. Throughout this process, there is so many time points where we are doing extensive cell count, cell viability testing, the purity of the product, and sterility testing. The last thing we want to do is use cells that are dead or not working or not healthy back into a patient that is diseased or needs help. We also don't want to introduce infectious disease. This is not so much when it's autologous, when the patient recipient are the same, but in a lot of these upcoming therapies, they're off the shelf or the donor is different than the recipient. And so we do not want to introduce an infectious disease to these are usually already immune compromised patients. As far as regulatory oversight, I can't talk about, or we cannot go into the mechanism of action and the excitement and hope of cell and gene therapies without truly understanding maybe the good players versus the bad players and regulatory oversight. So GMP is the gold standard for most of these cell therapies. And GMP stands for Good Manufacturing Practices. It's regulations designed by the US FDA. And we have multiple laboratories clinics at Mayo Clinic that are GMP certified. Now, in these private companies that say they're doing regenerative medicine or what have you not, they often do not have GMP facilities and they often don't even know what that is. And so just by you knowing this term, know this exists, and knowing this kind of validates whether or not the product you're working with is legitimate will help you a lot in the future. And so if a person in theory is saying that they have a product, it's a cell therapy product, it can regenerate something, blah, blah, blah. If it was not manufactured in a GMP regulatory oversight facility, that person can go to jail or prison. So it's very important that you know this is like kind of the gold standard, what we all go by. You also can see, and I really want you to focus on this image, these individuals are in bunny suits and they're in a sterile room. This room, you have to enter a GMP facility, oftentimes you have to enter 3 rooms to get to the final sterile room. And we know about to every particle that enters that room. So, and also all the infectious disease standardization and protocols are very intensive, more than you see in a surgery. And so you can just see by the imaging the differences kind of between developing a cell therapy product than developing an orthobiologic. Another regulatory, this is probably more in depth than you need to know. I just want you to know that it's two pathways in case you do want to get into this field and perform clinical trials, so you have some sort of base. The FDA and the Center for Biologics have two pathways for regulatory oversight. We have a 361 and a 351. What you need to know really is that 361 is for minimally manipulated products. And this is commonly what we have in bone marrow transplant, where we're using HPCs, hematopoietic stem cells, in a bone marrow transplant to have that curative effect, hopefully, in a patient that has a disease, hematological disease. And so the 361 is minimally manipulated, meaning we're not going in and genetically engineering the cell, and we're going to replace it back into the patient with the ideal same function as it had previously that we took it out of the body. 351s are the biologics license application and pre-market approval, so the BLA PMA pathway. And this is for all manipulated products, meaning that we're genetically modifying product. We're combining the product with a drug, device, or biologic. These are higher risk products, and there's more design control, regulatory oversight than products in the 361. Orthobiologics are interesting because the FDA sometimes doesn't know where the orthobiologics land in this world. So when there are legitimate clinical trials out there, there is always discussions on where biologics are, 361s or 351s. But just knowing that exists, I think it's important. Also, we're probably all very familiar with clinical trial phases, phase 1, 2, and 3. But maybe you're unfamiliar with INDs or BLAs. And this is something that I wish we were taught more about, even as residents and fellows. But an IND is something that is required if you made a novel therapeutic or you're an investigator in a clinical trial. Let's say industry approached you and they want to use this cool novel therapeutic. They had to submit for an IND for that to take place. And so being familiar, you may not ever submit an IND in your life, but if you're using a product that's new on the market, that's not a device, it requires them to submit an IND. So an IND is an investigational new drug. It's necessary to be submitted by the investigator or sponsor that directs a product involving unapproved drugs or therapies to the FDA. There's 3 types: commercial, research, and emergency. If you get anything out of this talk tonight, I want you to know about the emergency IND. So the emergency IND is for patients that have risk for high mortality very quickly. The FDA does not define what that means. So you can have a patient that you think has a couple months left in their life, has exhausted all treatments, and 2 weeks, 1 month, etc. But somebody that is a high risk for passing away. So the emergency IND is for any patient that you think that novel product, cell or gene therapy, medication, anything that is not approved for that indication, you think would work for your patient. So let's do an example. CD19 CAR T, commonly, and it's approved in multiple myeloma right now. Right now, they're using CD19 CAR Ts for MS and stiff person syndrome. If a person walks into your clinic, and before this time in this clinical trial world, where now we're using it for autoimmune diseases, the CD19 CAR T therapy, if you had a person walk in and they have MS, and you're thinking they're at the end of the road, and they have a risk for high mortality within the next year or so. You could submit an emergency IND to try a CD19 CAR T therapy that's already approved for multiple myeloma for that patient. So this is very important for your future going forward. If you see things on the market that you think that target could be used in a different patient population or a patient of interest that you are taking care of, you can submit this. And usually the FDA gets back to you within a week to 2 weeks, whether or not they approve that to go ahead with you to try that last resort drug for cell and gene therapy. Then after you get accepted to do an IND or IND submission, it's accepted commercially or research-wise, then you can get into your clinical trial, your phase 1, phase 2, phase 3. Obviously, the clinical trial, if it's an N of 1, it would not be a phase 1, phase 2, phase 3, but this is only for commercial research use. Once you get around to phase 2, phase 3, that's when you hear the biologics license application submitted. If you follow on LinkedIn, like any biotech or pharma companies, and they say we just applied for a BLA, that means that usually they have good results in their clinical trial. It has efficacy and safety profiles good. Usually that's what that means. And they are preparing to get it FDA approved and ready to distribute it across state lines. So that's also a big term to know going forward is looking out for new cool therapies. They're very open, these biotech and pharma companies, about when they submit their BLA. And so once you see that, you can have a light go off and say, oh, this therapy may be on the market pretty quickly, so I should be looking into it. All right, so a lot of this stuff, especially older attendings, I find, in younger ones, I shouldn't put them in a box either, or be biased, but a lot of people think this is, futuristic cell and gene therapy world, specifically in the neuro and musculoskeletal world. And I always like to say we have 12 approved on the market for our patients. And a lot of people don't even know that exists. So these are just a small list of what's on the market that we often see in PM&R clinics. And take it as you will, just knowing that they exist will be super important going forward, and advocating for access to your patients. But these are already FDA approved, they are on the market. These are the upcoming products that are showing really good results in clinical trial phase 2 and phase 3. I will say Dermacell, it's a cardiosphere-derived cell exosome therapeutic, is showing pretty good results for Duchenne muscular dystrophy, functional, upper lower extremity function, and cardiomyopathy. Kyverna is a CAR T therapy for MS and stiff person syndrome, and that's also showing pretty good efficacy. We also have an iPSC-derived therapy from multiple pharma and biotech companies within Parkinson's disease. Most of those are in the phase 3. IP is pretty shut on that, so I'm not necessarily sure whether or not that efficacy is good or bad with those trials, but knowing that they exist, they're in their phase 3, so they're getting closer and closer to the finish line. Also, various gene therapies, you're probably aware, you keep seeing that on your feed is, this child had a rare disease N of 1 and they had their gene therapy, it reversed their pathology. And those are on-demand manufacturing. So those are like INDs, N of 1 emergency INDs. So those aren't usually going to trial. They're just on-demand manufacturing for that one patient. There's also several trials with Rett syndrome going on right now, muscular dystrophy, of course, and then also seizures and epilepsy using human stem cells that release GABA for seizures. Now let's get into the orthobiologics. So probably you're more familiar right now clinically with orthobiologics. It seems sports medicine, pain, spine. They seem to promote orthobiologics quite a bit. And in PM&R residency on your rotations, you probably will even have the opportunity to perform orthobiologic procedures. So now that we discussed all that regulatory oversight, maybe a little bit boring, but very important to understand and know the terminology, orthobiologics is a whole different world. So if cellular therapies are heavily regulated and require years of clinical trials, millions to billions of dollars in research to make it to market, why are we performing unapproved orthobiologics procedures in clinic, and how did we go through the loopholes to make that happen? I will say there's several loopholes within orthobiologics world, but a lot of these loopholes exist with medical devices as well. So that's why you see a lot of cool new medical devices out on the market, and that's why a lot of companies invest in medical devices, because there's a lot of loopholes and less regulatory oversight to prove that the product is doing what it's supposed to. In the cell therapy world, the gene therapy, you have to be 100% accurate every time to get it approved. For the medical device world, it's a little bit more of a free-for-all. But in the orthobiologic world, so this is like a PRP, like centrifuge machine kit example. And as you can see, if you remember that picture I showed of the GMP manufacturing the cell therapy, it's very different. As you can imagine, this centrifuge is just on your clinical table in the room with the patient. So it's a very different manufacturing or creation of the product and standardization. But these devices are not FDA approved. They are 510K clear devices. And just to say that again, this does not mean that they're FDA approved. For 510 clear devices, it does not require regulatory oversight or validation at all that it works. And then, interestingly, studies involving existing orthobiologics commonly used the loophole of the same surgical procedure exception. So this means that with a biologic, we can use like these PRP machines if we're using it in the room of the patient. So the number one thing is it requires the removal and the implantation of the cells into the same patient from whom it was removed. So the donor and recipient are the same, and we're in the same surgical room that we are getting the cells from. And then the cells are in their original form. We're not manipulating and we're not doing any testing. So that is probably the biggest loophole that is out there for why we are able to provide orthobiologics clinically right now without, all this regulatory oversight and validated clinical trials. I will say, if you are interested in performing legitimate clinical trials in this space, which are extremely needed, it would likely require the same pathway as the cell therapy. So even though orthobiologics are not FDA approved, but they are often 510 cleared down this pathway of the same surgical exception, in a legitimate clinical trial, they would still need to go down a cellular therapy route, regulatory oversight, to be legitimate. So you'd still have to apply for that IND, investigational new drug. And if you have a product that you want to market, you would have to apply for a BLA. I do have an asterisk, very important, something that I do not see often in orthobiologics discussions. Biologics, the orthobiologics like PRP and BMAC, often contain very little amounts of progenitor stem cells, extremely little. Meaning that even though orthobiologics could provide certain growth factors, stimulate growth, stimulate certain cytokine release, that could help with regeneration, they are not regenerative. And they do not qualify as a regenerative medicine like a cell therapy world would. Because in cell therapy, we're validating that we have 100% progenitor stem cells in our product, ready for regeneration. So very different kind of thoughts. And if you go back to like the mechanism of action, We're in that orthobiologic. We like it for its growth factors. That's important. But we're not in that we're curative regenerative medicine space right now. So what's needed for orthobiologics to come to the playing field like CAR T has come for our patients? We need standardized clinical trials. We need legitimate physician scientists being interested in this and industry partnerships. And this is where things have gotten a little bit fishy, is because of this 510K same surgical exception. Why would an industry put millions to billions of dollars if clinicians can already do what they want? They can already do PRP. They don't need to validate. They don't need to do cell counts. They don't need to do infectious testing for these products. So why would you go back, and why would you spend all that money to invest? And as you can see going forward with any kind of novel therapy or medication, sometimes that is the issue, is that we went too fast and now we can't go back. But there's hope still. And then also standardized cellular content and dosing. So to make it more evidence-based, to make it an accepted procedure that would be reimbursed from insurance, and for other clinicians to be excited about it and want to refer their patients specifically for it, we really need to know, what are we injecting back into the patient? And how much do we want to inject? What is the dosage? How many cells, how many platelets are there? How many white WBCs are there? Also with potency assays, how are these cells even working? We could have a verification that we have so many millions of WBCs, but if they're all dead, or half of them are dead or not healthy, then what does that matter? And then validation testing, of course, and also infectious disease. That's a huge difference between I see in the cell therapy biologic world, is that even in cell therapy, a lot of our therapies are autologous, meaning the donor and recipient are the same. We're still doing intensive infectious disease testing, even though it probably may or may not matter if something comes up positive, because the recipient donor is the same. However, we don't do that all in biologics. And we are injecting cells. So that would be an important step for the validity of this world. So in summary, just a small chart I made to kind of give an overview of the difference between orthobiologics and cell therapies right now. Orthobiologics are not engineered or manipulated. Cellular therapies often are. The time to development, as you probably all have witnessed, are minutes in the centrifuge in the clinic, versus cellular therapies on average take between 2 to 6 weeks to manufacture, with many different specialists involved: pathology, hematology, medical directors of laboratories, and all the amazing techs and engineers involved. Sterility, viability, purity of the testing, we don't do with biologics right now. Cellular therapies, we do that on every product. FDA regulations, we just have that 510K clearance designation. I will say, before this last year, there was a lot of discussion about maybe we shouldn't even have a 510 clearance designation anymore, because that leads to issues kind of like what we're seeing with this, but also with medical devices, especially constantly putting out medical devices that may or may not work, but they're able to get through that with 510K clearance. And then cellular therapies, of course, we have to have that GMP facility. That's billions of dollars to even create. We have to go through the 351, 361 approvals, and then all the many, many accreditation bodies, which I didn't touch today or discuss. And then orthobiologics, that progenitor regenerative medicine type cell that we care so much about in this world. Orthobiologics usually are very minimal. Cellular therapies, we can manipulate it to be up to 100% the product. So I really see great value in the current use of orthobiologics. I really do see the future of being very hopeful and what we could provide to our patients in this unmet need. It's pretty exciting, especially because of all the growth factors and cytokines that biologics can release. But it's really essential to exercise caution when using terms like stem cells, regeneration, progenitor cells when advising patients. A solution to this all, and hopefully one of you will develop this someday, is to have an off-the-shelf GMP manufactured allogeneic MSC or orthobiologic. So we can truly say we are potentially regenerating something in the joint or wherever we're injecting or infusing. And we can help patients with neuromuscular skeletal disorders, not just sports med, because there's a lot of unmet need with our patients. Now, if you're interested in this world, what are the education opportunities right now? So I have a list up here. There's multiple available pathways. Currently, there's 4 fellowships like mine that focus on the actual development, manufacturing, translating cell and gene therapies, and then also providing access to patients for approved therapies on the market. So Mayo Clinic has one, UPenn. UPenn is called the Silicon Valley because that's where most cell and gene therapies have come out of. We also have University of California, San Francisco, and University of Minnesota. Additionally, there are 2 gene therapy programs nationwide, OSU, and then the University of Utah. And then I do just want to focus, and I know if you're going into PM&R, this probably wouldn't, you qualify for, but there's over 20 fellowships for clinical hem-onc physicians for cell therapy. I put this on the screen because there is a huge need for us neuro MSK specialists to be in this world because they have been very focused on hematological cell and gene therapy access and indications. But there's over 4,000, some say over 10,000 cell and gene therapies in development, and around 46% of them are neuro and MSK. So you think of all these coming up in our time, which is great, but who are going to feel confident to prescribing this for these patients? Who are going to feel confident on knowing the risk versus benefit profile and the surveillance after infusion or giving these therapies to these patients for lifespan? So it's really, I think, in our hands in the future because Hem-Onc has done a wonderful job about staying on top of novel therapies, and they have a lot more access to their upcoming physicians for education, but there's just something to consider. And that is all I have. So I will take any questions.
Like I said, guys, feel free. You can put them in the chat or you're here to go on while we wait. Holly in the chat says, can you speak to the validity of varying efficacy of medical devices and processing techniques with orthobiologics, specifically related to the financial incentives due to the lack of ability to patent autobiologics?
Yeah, that's a great question. So I think it all comes back to the lack of standardization. And right now, since it's just taken a form of its own and clinically, pretty much everyone in the space knows that it doesn't matter if you did PRP for the same person each day, that product is always going to be different. And so until we have a product that is completely standard and that we know exactly what it is, it's usually not going to be like IP worthy, I guess. The FDA is very specific. Each country is very different in how they, or I shouldn't say very different. Some are very similar and some are very different on how they regulate IP and what's approved, what's not. The US, the FDA is very specific that you have to know your target and you have to know the mechanism of action for it to be approved. And so unless we have the legitimate studies of this, I don't foresee that being approved. Now with this administration, I don't know, but to know the legitimacy of it is important. But yeah, it's kind of complicated, I'd say, but it's very ingrained in the legal world and fine print world. But just knowing the mechanism of action and target, number one thing when you're making any product. And unfortunately, with the biologics, we don't have that answer. And we know for a fact we can't, right now, as it stands, we can't standardize it even with the same patient. So that would make it very difficult to get it approved, which is why it'd be great to have a donor supply and off the shelf because we know exactly what we're getting.
I had a quick question, actually.
Yeah.
My name is Michael. I'm a third year medical student. Thank you so much for your talk. I thought it was really, really informative. The one thing that I was in sort of getting interested in was this whole 510K clearance
Yeah.
with the orthobiologics, and that's the workaround with it. You had mentioned something along the lines of it might actually be helpful to remove it so that they're sort of forced to do the dirty work and figuring out, like cell counts and all those things. Do you
Yeah.
think in the long run that is something that is actually good, or is that something that we should stay away from just because it provides enough of a benefit now? Or do you think overall it's something that would just wind up hurting people's, uh, I guess, psyche more than anything else? But
Yeah, no, I mean it's very complicated. I could say I can make an argument for either way. But I think from what I've learned from the standpoint on what makes a successful drug, what makes buy-in from different specialties and how different specialists view each other, I think we need to do the dirty work and go back. Um, but with the limitation to say there is something positive that we're seeing and it may work. And especially with PRP with NEOA, I think that's pretty much standard that that does something positive for those patients. Um, but being, I guess, humble in the sense of like, we haven't done the correct research, but we do know there is help and we can work around that right now with the regulations with the 510K. So we can provide it for people who are socioeconomically want to try it because it's out of pocket. But yeah, no, I think regardless, we have to go back and we have to do the work we need to do if we want to get the buy-in, especially from industry. Industry is now like, I'm not investing in actual clinical trials for this.
yeah,
And,
yeah,
and we need industry. We can't do it academic alone, you know. So
yeah, no, that's completely fair. Because in my head, I was thinking like, oh, like maybe it would be useful to do research on like, do we need to dive deeper into this? And then in my head, I'm like, but that's already research. Like, we already that already is the hard work that you're doing. But that makes sense. I appreciate that.
it is complicated. So yeah, every opinion is valid, except the opinions that are like, I'm a regenerative medicine and I'm regenerating and there's stem cells all in this product. It's like, sure, maybe there is, but you got to validate that. Like, I'm not, no, in no medical profession can you say you're giving this pill and tell me you don't know the mechanism of action, you don't even know the name of it, but you feel that it is made of something that it may or may not be. So, yeah, there's no feel like that.
Completely fair. Completely fair.
Holly put in the chat, do you see many companies filing for orphan drug status to fast track any of these specific cell therapies?
Yes, lots. Yep. I didn't go into that. You're amazing. Who's that, Holly? To know that that exists. Hello, Holly. Great. You know your stuff. Yes, no, there's a lot of different designations right now. RMAT is a designation, orphan drug. There's, I think there's 4 to 5. You may know better than I, but there's multiple designations. And yes, most everybody tries to get one of those designations. It's very lucrative. Uh, and for those who may not know about designations, so designations is another thing to you could add to these industry or academic profiles. If you're developing a product, you're wanting to get to the fast to the market, to the finish line with like the least amount of cost, because a lot of these cost between $1 million to $1 billion to even get from basic to market. Um, to recoup those costs, you can get fast tracked to approval, and it can kind of help with how much they've invested. Yes. And is that answer? Okay.
Yeah, thank you. I appreciate it. Prior to medical school, I actually was looking more at the intersection of medicine and business, and I did go-to-market strategy for one of Mayo's neuroscience patent holdings, and
Awesome.
the the patent holder was very adamant in it getting to patients, making sure that the patent didn't get shelved, but it looked like the most legitimate pathway to development was going to be through orphan drug status. So, and I don't know if you recall, but for everyone on the call, orphan drug status, generally the drug or therapeutic has to help
Less than
patients with,
2,000.
yeah, thank you.
Yeah. Yeah, orphan drug status. That's why, too, sometimes you'll have a patient come into your clinic and they have like extremely rare disease, maybe N of less than 10. Do not think that therapy does not exist. They're more likely at this point, because of all the lucrativeness with having an orphan drug or rare disease designation, to have a therapy in the works than somebody with like a very common disease or disorder. And so you'll see that a lot with rare disease, especially coming up more and more, because they can recoup those costs of all the like you can develop like $1 million, $2 million therapy that may help 10 people, but it was nothing essentially to produce that because you got to recoup it from all the like designations and reviews from the FDA. So
Thank you for your detailed presentation. It's really helpful to understand the differences and the pros and cons of both. I think there's a lack of education and awareness around it right now.
totally, yeah. And we should, I don't know why we don't get this education. I mean, I didn't get it until I got into fellowship. And I know most people don't at all. And so learning how to play the game so we can change the game is important.
I didn't get it until this presentation.
Awesome.
All right, I think we'll end on this last question, and this is a popular one that was sent in even before you came on, Dr. Strader, and it's from Bilal, and sorry if I'm mispronouncing your name. It says, can you share a little bit more about your current fellowship structure, what your day-to-day looks like, and what are you actively working on right now that's you're excited about that you can share with us? So yeah, take that where you want.
All right. Well, so most of the fellowships, cell therapy fellowships, are 1 year. Mayo is different. It's a 2-year track. Most of the fellowships right now are heavily focused on those hematology, CAR T type manufacturing. Mayo was different, which is why I chose Mayo, because I have had rotations on developing mesenchymal stromal cell therapies, dendritic cell vaccines, and additional like cell and gene therapies. So more novel therapies than your typical bone marrow and cell gene therapy or CAR T therapy. So last year I had a full year of rotations. And so I acted as the medical laboratory director when I was on call. So anything that was going on with the cells or during the infusion or toxicity with the patient was a call to me. So I was more the physician over the cell product, the living cells, than I was necessarily to the whole patient, equally important. But that's why a lot of these cell and gene therapies are within the pathology department, because pathologists are usually medical directors of laboratories, clinical laboratories. And so that's why it fits really well in the pathology world. But all my rotations were different based on the cell product that we were developing, or they were developing in the certain lab. This year, I am taking call as if I am the medical director, no longer like as much oversight, which is a very nice transition. And then I am focusing on, I have a clinical trial that has gotten funded. So I'm doing a pre-rehab trial. And my dream is to make a therapeutic, but right now I'm personally going through the trial at a very basic level to understand the ins and outs of developing a clinical trial. So what I'm doing in this pre-rehab trial is for CAR T. So I'm offering pre-rehab for or my team, not just I. It's a team of about 30. Uh, we are offering pre-rehab with the heme team and PM&R team, so very multidisciplinary. And throughout the process, we are using wearables, we're using an app, so it's decentralized, so they're at home performing the pre-rehab program, not in a clinic or not inpatient. And we are taking blood samples throughout, and I am extracting the extracellular vesicles to find a biomarker. So my dream would to be is to find a biomarker or therapeutic target to make a therapy. So that's right now what I'm doing majority of my days. Yeah.
That's really, really awesome. And we thank you so much, Dr. Strader, for the sake of your time and for everyone else's. We'll go ahead and end the meeting here. Thank you, everyone, for participating. And hopefully you guys learned a lot just like us and just like me, I should say. And Dr. Strader, is there a place that they can find you?
Yeah,
LinkedIn,
you
you're
can send
active
it. I
there.
should have said, yeah, you can send out my email. I have now.
Okay, that's
She's
very kind of you. Thank you guys, and hopefully you guys have a happy holidays.